Hello, so welcome back. So this is for the second part of the Plankton II lecture. I just wanted to start this off by we mentioned in the previous half of the lecture about Argo floats. This is actually just to show that these techniques are still being used. Here we have a series of photos that were taken from the cruise that I mentioned, which goes down every year between the UK and Antarctica. So this is a picture of them actually launching an Argo float in the night. You can see this very blurred yellow blob as it's thrown from the side of the ship. And this is them prior to them launching. So you can see them here. They're actually marking them externally, so on the outside. I think these particular ones were sponsored by schools to help with the research. Here's the float once it's actually put into the water. Another one being released during the day. And there it is a few minutes after release as it floats away at the surface. The other thing I wanted to show you was this is data from I think this year, from the cruise this year. So this is from November the 2nd of this year, so 5 days ago. You can see here this is actually a pumped water system. They were using this to study particles at depth, and you can actually see the impression here. This is a diatom plankton species. You can see the body of the diatom here, and then its two kind of antennae coming out here, which is where it's been sucked through the system and then caught on the filter paper. So I just wanted to show that before we then carried on with the lecture. Okay. So we're now going to talk we're going to have a case study, and then I'm going to talk a bit about distributions. So this is an example of how you can combine all of those different methods to actually study plankton distributions that we talked about. So calcifiers. So we mentioned right at the beginning that one of the groups of phytoplankton are calcifiers or coccolithophores, which are the ones I'm going to mention here. The coccolithophores are actually only about 30% of the calcifying plankton distributions in the ocean, but these happen to be the ones you can actually see from space as well. So this is an example of a coccolithophore cell, a healthy one that's living under an electron microscope. So you can see all these little platelets. These are called liths. They're like shields around protecting the actual cell. So this is when it's alive, and you can see when moving the cursor down, this one, the second picture here, is actually as the coccolithophore is dying. So as it dies, all of the liths, these shields, kind of peel open and flatten out, and they fall apart. And at this point, all of these liths act as mirrors, and they reflect light, which means that you can see them from space. So this is a picture as observed from space. It's from a satellite called MODIS, which is a NASA instrument. This is actually the north coast of Norway here, and this whole area of blue and turquoise you can see in the water are coccolithophores which are dying, and they're shedding their liths and therefore reflecting the light straight back up into space, and hence they can be seen. So what I'm briefly going to talk about are the results of two different papers or publications that are studying how the growth of these plankton can actually modulate the oceanic sink of carbon because they're unique and they calcify in the water, and they regularly form in the North Atlantic. And the question we were trying to answer is what is their impact in terms of absorbing how they change the absorbance of carbon in the North Atlantic. They can also be seen. So this example I showed was, in fact, sorry, if I go back, this is Norway, as I mentioned, north coast of Norway. They also form other places in the North Atlantic, including near off the UK coast. So this is one here off the edge of the shelf. This is obviously Cornwall coming down the southwest of the UK, and this is Brittany of France. This is a very large bloom. So you, for example, to see the size of them here in relation to the size of this bloom is actually bigger than the whole of Cornwall, the Cornish county. And they're having instances where they've formed, in fact, just off the coast. So they've been within an easy sail of Plymouth, for example. So when they grow, these coccolithophores, as with most plants, they take up carbon dioxide from the water, but they also uniquely, as calcifiers, emit. So they push out carbon dioxide into the waters. So the net result of their growth actually increases the amount of carbon dioxide in the water, and this therefore reduces the ability of that water to uptake more carbon dioxide from the atmosphere. And it turns out that this can have quite a large impact in regions where they grow. So for example, you can see here, this is the north of Norway, the example I gave a second ago. And as a result of their growth in the water and the fact they increase the amount of carbon dioxide in the water, they actually reduce the oceanic sink in this region by up to 55% during any 1 month. So you can see they predominantly form in areas in these subpolar waters. You get formations of them around Iceland, and there have been instances of them forming out into the middle of the North Atlantic. So it turns out overall, over a long time series, they can actually reduce the North Atlantic sink of CO2 by up to 28%, which is quite a large impact for one very small, tiny animal. Plant. So last of all in this lecture I'm going to talk about the distributions of plankton. So I showed you the global view as seen from satellite. This is another cartoon at one perspective of global spatial plankton distributions. And you can see straight away that the distributions are highly influenced by ocean physics. So plankton means drifter, so they're essentially at the mercy of ocean physics, and you can immediately see clear signals here. So for example, this is the impact of the Gulf Stream and the North Atlantic drift. So the Gulf Stream comes across and then it turns into the North Atlantic drift. Again, this is the Kuroshio current here, the equivalent in the Pacific, geostrophic current. And this one at the bottom here is the circumpolar current, the Antarctic circumpolar current. Again, clearly these are acting on the distributions and defining and shaping the spatial distributions of plankton. So the impact of physics. These are upwelling regions which we talked about, and then these are very low abundance areas where you have the gyres, which is the rotation, large-scale rotation of water as a result of geostrophic currents. So the one thing we should also note is that plankton concentrations, phytoplankton concentrations, are logarithmically distributed. By this I mean that they vary either from very, very small right up to very, very large, and the increase that you'll get is not linear. So you will get suddenly, if you start in the open ocean, you have very low values, and then as you approach this region, the upwelling regions, you've got a very sudden and exponential increase in concentrations. So that's what I mean by logarithmically distributed. So whenever you analyze these data or you plot them, you always will use a logarithmic scale. Otherwise you'd have to have a very, very large graph and it would be very hard to interpret. So there's been a lot of work looking at distributions and trends. These are more examples of how you can pull together lots of different methods and the advantages and disadvantages of the different methods. So there's this paper that was published in Nature in 2010 looking at global phytoplankton decline. They identified from 2 different data sets that globally phytoplankton were declining in abundance. So they had 2 different data sets. One was the in situ chlorophyll data, so these are in situ measurements which have been taken from ships, and they collated a large data set from all ship measurements that they could find. And they also used a transparency approach, which is using a Secchi disk. Just so I explain what a Secchi disk is in case you haven't come across it. Secchi disk looks a bit like this. So it's a black and white disk which you slowly lower through the water, and then you record the point at which you can no longer see the disk, and that becomes the Secchi disk depth, or it's an estimate or a proxy for the depth at which the light is no longer penetrating through the water. So you can see here that people are lowering the water, the Secchi dips into the water. So they used the Secchi disk data set, so they collated all the Secchi disk and Secchi depth data that they could find globally. And from these 2 methods, they then inferred that there was a reduction, a decline over the past century in phytoplankton distributions. So this is their map that they created in terms of different trends. You can see red is a positive trend, red and yellow, whereas blues are negative trends. And you can see predominantly they have a large number of negative trends, and there's two examples here that are shown at the bottom, one for the Arctic region and one for the North Atlantic. However, this piece of work was met with some discussion because not everyone actually agreed with these conclusions. So if we now look at, if you look at this area here, for example, around the North Atlantic, including the UK and European shelf, this area within this particular study showed that there was a negative reduction and trends in the phytoplankton distributions within this area based on the in situ chlorophyll and the Secchi depth data. However, this study, they responded to the previous work, questioning whether there was actually a decline in phytoplankton, and this is based on data that collected by the CPR, the continuous plankton recorder that I showed you earlier on in the lecture. And they split their data up into all of these different regions because they had a lot of trawls from all these ships pulling their CPRs, and they show that there was in fact an increase in abundance of particular types of plankton within this region over the same time period as the previous study. And this actually, this response meant that these two teams actually started working collaboratively to identify why and how the two different analysis methods had come up with completely opposing results. More recently, there's been some work. This is work that was published in 2015, doing something similar from space. And here they identified that it's a combination of different reasons. So those previous two studies effectively were both right, and it depends on the type of plankton you actually study and the different groups and species, because they all are showing different responses. So, for example, in our North Atlantic region, there are some variables which are showing a decline, such as total chlorophyll, which concurs with the 2010 study. But they also have identified that there is an increase in the amount of coccolithophores within this region, which is consistent with the second study. So that result is consistent with this Saphos-led study. So just to summarise for this lecture, there's many different types of methods for studying phytoplankton, from fine scale to global scale. They all have different advantages and disadvantages. The important thing is that you will invariably use multiple approaches to actually study phytoplankton. And the key important thing to remember is that you have to take into account the fact they all have different performance and the underlying assumptions for each of those different techniques. And clearly the examples I've shown at the end there, understanding the distribution of phytoplankton and how these are changing, is a challenging and complex issue. But it does require understanding of lots of different environmental systems and processes. So if you want to study plankton, for example, you also need to have a good understanding of ocean physics. And then if you want to study the impact of the plankton, then it may well turn out you need to have an understanding of exchange between the ocean and the atmosphere, and therefore this requires some understanding of chemistry. So all of this work is clearly interdisciplinary. There's some suggested further reading here. This paper here is the one I mentioned at the end, where they're looking at decadal trends in phytoplankton distributions from space. There is also this second paper which talks about how you can use size detection from space as well to identify different functional groups. So a similar process to what the flow cytometer was doing, but actually how you can apply that kind of approach to space observations. This last paper here is fairly old, but however it's a nice, short, snappy study on how you can use coccolithophores for actually as an indicator of changing climate. And indeed, you'll find that the results from this paper, or the proposed hypothesis or suggested reasoning for the changes they detect, are actually consistent with this more recent work where they identified that the coccolithophores were in fact, or have in fact, increased in abundance in the North Atlantic. So you'll see that these two pieces of work follow on nicely from each other. And also at the bottom here there's a TED Talk which talks a bit more about the secret life of plankton, which you may find useful and informative as well. Okay, so thank you very much for listening, and that's the end of the lecture.